Welcome to This Explainer. Today, we're taking Dr. Mukesh Pimpleskar's incredibly detailed clinical notes and transforming them into a high-stakes medical detective story. We're going to uncover exactly what happens in a microbiology lab, step by step, from the very moment a sample is collected all the way to that critical final diagnosis.
Okay, let's dive right in by addressing a mystery we've literally all experienced at the doctor's office. You hand over a little plastic cup, maybe a swab is taken or a blood vial is drawn, and then, well, it just disappears behind closed doors, right? What actually happens back there? It turns out it's a rigorous, highly structured investigation.
Look, the crucial point here is that this isn't just routine paperwork. A compromised crime scene, meaning a poorly handled sample, can lead to catastrophic medical consequences. If the lab gets a false positive or a false negative, it completely throws off the antibiotic selection. That means treatment failure for the patient, and on a global scale, it fuels something so much worse.
And this brings us to the ultimate villain of our story, antimicrobial resistance, or AMR. This is a genuinely dangerous phenomenon where pathogens mutate and survive our absolute best medical treatments. And guess what? It is actively accelerated when the wrong antibiotics are prescribed. That is exactly why following stringent, internationally recognized laboratory guidelines like CLSI or EUCAST is absolutely essential. The lab is literally our frontline defense against AMR.
So here is our investigative roadmap. We're going to cover the crucial first step, take our first looks with direct microscopy, grow the suspects, interrogate the microbes, test our weapons, and finally, deliver the verdict.
All right, let's kick things off with Section 1, the crucial first step, which is all about securing the scene.
The really fascinating thing here is how these 5 rights ensure we lock down the scene perfectly. Think about it. If you're drawing blood to check for septicemia, you absolutely must get it from the right patient at the right time, which means collecting it before any antibiotics are even started. You have to use flawless aseptic technique, like disinfecting the skin with chlorhexidine. Or if it's a urine sample, ensuring a midstream clean catch. Improper collection just invites contamination from everyday bacteria, totally throwing off the entire investigation.
With the evidence flawlessly collected and transported, we move into the lab for Section 2, First Looks, where we start spotting the suspects using direct microscopy.
Now this is where it gets really cool. The Gram stain. This incredibly vital test was developed all the way back in 1884 by Hans Christian Gram, and it divides our microscopic suspects into 2 distinct physical profiles based entirely on their cell wall structure. Gram-positive bacteria, like Staphylococcus, have a thick wall that traps the stain, glowing a deep purple. Meanwhile, Gram-negative bacteria, like E. coli or Salmonella, have a thin wall and show up pink or red. Boom! In just minutes, the doctor already has a massive clue about what they're fighting.
But the lab doesn't stop at the Gram stain. They use an entire toolkit of specialized rapid visual tests to identify unique suspects. Looking for tuberculosis? They'll use an acid-fast stain, where the tough mycobacterium appears bright red against a blue background. Or if they're looking for a fungal invader like Candida, a potassium hydroxide, or KOH mount, is used because it literally dissolves the human tissue in the sample, leaving only the fungal structures intact for the detective to see. How wild is that?
Once we have that preliminary visual, we move right along to Section 3, growing the suspects to create a proper lineup.
So, to coax these suspects out of hiding, we have to recreate their absolute ideal living environments. And honestly, that varies drastically. Bacteria generally thrive right at human body temperature, around 35 to 37 degrees Celsius, and pop up in just 18 to 24 hours. Fungi, on the other hand, prefer it a bit cooler, around 25 to 30 degrees, and might take anywhere from 3 to 5 days or even longer to finally show their faces.
Temperature alone isn't enough, though. Culturing requires highly specific diets. There's a stark contrast between general-purpose media and selective media. Take blood agar, for instance. It's an enriched buffet that grows a lot of things and reveals if a bacteria destroys red blood cells, a process called hemolysis. But then you have McConkey agar. This is a selective medium. It cleverly acts like a bouncer, filtering out the Gram-positive suspects entirely, only letting Gram-negative bacilli grow, and even color codes them pink if they ferment lactose.
And for the really tricky culprits, the lab deploys these highly specialized environments to force them to reveal their true identities. Fastidious organisms, basically the really picky eaters of the microbe world, get chocolate agar, which contains cooked lysed blood. If we're looking at a urine culture, we use CLED agar to stop certain highly motile bacteria, like Proteus, from swarming all over the plate and ruining the evidence. And for fungi, they get their own dedicated real estate called Saborod dextrose agar.
Moving into Section 4, interrogating the microbes. With our pure colonies isolated, we bring them straight into the interrogation room for biochemical testing.
This is where we literally force the microbes to confess through chemical reactions. Take the catalase test, for example. We add hydrogen peroxide. If it aggressively bubbles, the microbe produces the catalase enzyme, and that points straight to Staphylococcus. No bubbles? It's Streptococcus. Or look at the indole test. E. coli tests positive, while Klebsiella tests negative. These biochemical tests corner the microbes until they tell us exactly what species they are.
Next is Section 5, testing the weapons, also known as Antimicrobial Susceptibility Testing. We know exactly who the culprit is now, so the next crucial step is finding out which weapon can actually take them down safely and effectively.
Let's look at how the Kirby-Bauer disk diffusion method builds our defense. First, the lab makes a perfectly concentrated liquid suspension of our suspect, matching it to a visual baseline called a 0.5 McFarland standard. They swab this all over a specific agar plate to create a totally uniform lawn of bacteria. Then they drop little paper discs, each soaked in a different antibiotic, right onto the plate. After incubating it overnight, they inspect the battlefield.
The physical measurement of the damage dictates the exact future of the patient's treatment. Around the effective antibiotic discs, there will be a clear halo where the bacteria couldn't grow. The lab measures this zone of inhibition in millimeters. Based on super strict guidelines, they grade the drug. Sensitive means it works at a normal dose. Intermediate means it might work at a high dose. And resistant means the bacteria simply ignores the drug. If the doctor prescribes this, the treatment will outright fail.
Finally, we reach Section 6, the final verdict. It is time to close the case. All the clues have been gathered, tested, and analyzed, bringing us to the conclusion of our investigation.
The final laboratory report isn't just a basic summary, it's the ultimate case file. It carefully integrates everything. It confirms the specimen quality, details the microscopy findings, identifies the exact organism biochemically, and most importantly, provides that crucial S, I, and R antibiotic susceptibility pattern. This entire document guides your doctor's exact personalized treatment plan.
But hold on, how do we know we can trust this entire process? To ensure absolute accuracy in this life-or-death environment, the lab constantly tests itself using internationally standardized quality control strains. They run known strains of E. coli or S. aureus with specific ATCC identification numbers through their own tests. If these known strains don't react exactly as predicted, the lab knows their setup is off and they halt testing to fix it. It's a brilliant failsafe.
So if there's one crucial point to take away from Dr. Pimplescar's workflow, it's this incredibly simple but profound equation. Quality specimen equals quality diagnosis. Every single high-tech stain, specialized agar and biochemical interrogation relies entirely on that very first step of you or your nurse providing an uncontaminated, properly collected sample. I'll leave you with this question as we wrap up this explainer. The fight against antimicrobial resistance isn't just happening in massive pharmaceutical research labs, it's happening every single day in your local clinic's microbiology department. I invite you to appreciate the rigorous, unseen medical detective work happening every time you hand over a sample. Thank you so much for diving into the fascinating world of the lab with me, and keep asking questions about the hidden science all around us.